Greetings, everyone. Today, I'm going to present you the effect of sex and habitat-dependent ecological variables on the vertebral stripe distribution in the endemic water frog, which is Pelophylax cypriensis in Cyprus. This is the introduction part. The vertebral stripe is a common pattern in many distantly related aneuron amphibians around the world. The vertebral stripe of the frog is thought to offer the substrate matching or disruptive camouflage, although its origins and selective pressures are unknown. This would prevent the recognition of the animal's body shape by visually splitting it into two parts. You can see that in the figure right into your slide. I took the photos of the frogs which have a dorsal pattern and represent it to you. These phenotypes shared across divergent lineages can result from ancestral alleles conserved over millions of years of evolution, or they have evolved independently multiple times. Although predator-mediated selection is a widely assumed mechanism for the evolution and maintenance of most color patterns, the relationship between the vertebral stripe and the survival is not known. The Cyprus water frog, Pelophylax cypriensis, is an endemic species of water frog that is found mostly in the humid areas of Cyprus. As in other eastern Mediterranean region water frogs, this endemic species shows morphological and habitat variation. There are many studies that are done about the vertebral stripes in other amphibians, but there is not any data regarding water frogs. In this investigation, our aim is to add some data that is considerable for literature. Water frog Pelophylax cypriensis is one of the target species in such isolated islands. And the aim of this study is to investigate the relationship between the environmental variables and the striped water frogs, because it is still unknown whether shared phenotypes across different frog lineages are the result of ancient alleles or the influence of development and a variety of environmental conditions. The aim of this experiment is to study the relationships between the vertebral stripe patterns, which are striped, slightly striped, and unstriped, that are shown in Figure 1 respectively, and habitat-dependent ecological variables, which are the habitat types, water fluidity, such as standing versus non-standing, and the levels of salinity. And the second aim is to investigate the relation between the sex and ventral stripe patterns. This is the material and methods section. I did the sampling all by myself, and I collected 78 individuals from 26 different localities. I photographed them and recorded them by using and drawing a locality map of Cyprus. After the data record, I released the samples into the natural environment without harming. And this is the map that I did, which shows the sampling localities throughout Cyprus in both north and the south of Cyprus. For data collection, the ventral patterns are classified as striped, unstriped, and slightly striped. We collected the sex, which are male and female, and there are also habitat-dependent ecological variables, such as habitat types, water fluidity, and salinity. For the data analysis, the histogram graphic was drawn in RStudio version 1.3 in order to look at the habitat preferences of our water frog, Pelophylax cypriensis. To investigate the relationships between the vertebral stripe patterns and habitat type, water flow, salinity, and gender, by using the SPSS program, we used non-parametric method of Kruskal-Wallis test. And the p-value is accepted being less than 0.05, which shows a significance level in statistical analysis for all the hypothesis tests. These are the results section. Many graphs were drawn in order to investigate the relationship between the dorsal pattern and the habitat preferences or the gender. For example, the first graph describes the relationship between the habitat preferences of the water frog species. As you can see from the graph, they preferred to live in the F stream the most. It shows that they are highly variable in terms of habitat selection, such as in Anatolian borer frogs. In the second graph, we are investigating the dorsal pattern through habitat types. As you can see that the blue one, which shows the slightly striped individuals, they are only found in artificial ponds. Striped individuals are mostly found in dams, lake, marshes, stream, and borer channels. And unstriped individuals were found in all types of habitats. They prefer all types of habitats in order to inhabit them. But as you can see from the Kruskal-Wallis statistical test, the p-value is 0.005, so that we are rejecting the null hypothesis. In this graph, we are investigating the relationship between the vertebral stripe patterns and borer flow. Null hypothesis is rejected because we accept the null hypothesis in which they have a p-value being less than 0.05. So that we can say that there is a statistical relationship between the vertebral stripe patterns and borer flow. However, as you can see that the exceptional value of the slightly striped individuals were only found in stagnant borer, whereas the other types of both striped and unstriped individuals were found in both non-stagnant and stagnant borer. In this graph, we are investigating the dorsal pattern through water salinity level. We can see that null hypothesis is accepted, so that we can say that there are no statistically significant relationship. However, the blue ones, which are the slightly striped individuals, were only found in high salinity habitats. In this graph, we are investigating the relationship between the vertebral stripe patterns and sex. The p-value is 0.52, so that we accept the null hypothesis. And we can say that salinity pattern does not affect the dorsal pattern, so that there is no significant relationship between the vertebral stripe patterns and the sex. However, slightly striped individuals are exclusively male, and subadult animals were completely devoid of stripes. To conclude our research, we can see that there is a statistically significant relationship between the vertebral stripe patterns, habitat type, and borer flow, which suggests that these environmental variables may have an impact on the development of the vertebral stripes. The vertebral stripe pattern, on the other hand, did not significantly correlate with either sex or salinity. However, to conclude these results, more investigation showed that slightly striped individuals were only found in high salinity stagnant water habitats and that they were exclusively male. Furthermore, subadult animals were completely devoid of stripes. These findings imply that the observed variation in vertebral stripes within these species appears to be closely associated with habitat-dependent ecological variables. And the results of the experiments show that these findings are important to see which environmental factors could act on the development of such an evolutionary important vertebral stripe trait. In addition to this, more data is needed from distinct species in order to reveal the evolutionary development of this trait. This is the acknowledgement section, and the study was supported by TÜBİTAK 2209A. Thank you for listening.